speaker is Professor Andrew Owen from the University of Liverpool, and he will speak on pharmacokinetic evaluation of antiretroviral solid drug nanoparticles. Please.
Thanks very much. So I'm going to share with you some of our first-in-human data for a project we've been working on for 5 or 6 years now. Just say that our formulation strategy, our preclinical data, is all summarized in these 2 previously published papers. Our overall strategy is to try and augment the pharmacokinetics of antiretroviral drugs by formulating solid drug nanoparticles. And a summary of the preclinical data is shown here, and you can see the magnitude of pharmacokinetic enhancement that we get in our physiologically based PK modeling, and a similar effect was also seen in preclinical species. We think that this approach has a lot of potential. A lot of potential advantage for pediatric formulations. Currently, antiretroviral drugs are given to very young children in very high amounts of organic solvents. We can formulate directly into water, and so there's a putative benefit there for pediatric applications. However, the data I'm going to share with you today was healthy adult volunteers dosed with powder-filled capsules. So our aims were to investigate the pharmacokinetics of the lower dose lopinavir and efavirenz solid drug nanoparticle formulations in healthy volunteers. We constructed population pharmacokinetic models to describe the available data and then use those models to simulate against currently clinically used formulations. And we also investigated multiple dosing safety and tolerability of these formulations. So the clinical strategy was slightly different for lopinavir than it was for efavirenz. Lopinavir is given to patients in combination with ritonavir to boost the pharmacokinetics by inhibition of cytochrome P450-3A4. So the data I'm going to show you is for a 100 milligram twice daily dosing of the nanoformulation. We dosed up to steady state. I'm just going to share with you the tail pharmacokinetic profiles at the end of the dosing interval. For efavirenz, we opted to use a lower concentration. The efavirenz was dosed at 50 milligrams. And again, since this is a long half-life drug, I'm going to share with you just the tail study at the end of the final dose. So for the population pharmacokinetic modeling, the data was input into NOMMEM. We constructed structural models, either 1 compartment for lopinavir or 2 compartment for efavirenz. We did a number of model evaluations around the POP-PK model. I'm going to show you the visual predictive check. And once the model development was complete, we then went into model application. We conducted simulations with these models in 1,000 virtual healthy volunteers. And we used these simulations to compare the pharmacokinetics for the nanoformulations versus the pharmacokinetic data which were already available from patient studies, and express this as a geometric mean ratio for the nanoformulation versus the conventional formulation. So this slide just summarizes the lopinavir model structure. Obviously, we had to model the ritonavir booster, pharmacokinetic booster, in addition to the lopinavir itself. They were both 1 compartment models. but we incorporated an indirect response model within this to capture the variability in the ritonavir concentrations and the impact of that on the cytochrome P450-3A4 enzyme pool within the liver, and used that to capture the impact of that on the lopinavir pharmacokinetics. The model summary is shown on the right here. I'm not going to go through that in detail. And the individual pharmacokinetic profiles for the 5 healthy volunteers dosed with the lopinavir nanoformulation is shown at the bottom of the slide. So for efavirenz, we had a 2-compartment model, which meant we had an additional parameter to capture the intercompartmental kinetics. And again, the model parameters are summarized on the top right here, and the individual pharmacokinetic profiles for the 4 healthy volunteers is shown at the bottom of the slide. So this is our visual predictive check for model validation. The red lines here show the median pharmacokinetics for the different drugs. The black dotted lines show the 90% prediction intervals. You can see for the lopinavir solid drug nanoparticles, 94% of the observations were within the 90% prediction interval. For the ritonavir, which was dosed as a conventional formulation, so the ritonavir wasn't nanoformulated, but for the ritonavir, the 96% of the observations were within the 90% prediction interval. And for efavirenz, 92% of the observations were within that interval as well. So moving on to model application, this was the simulation of a 200-100 milligram dose of lopinavir in the nanoformulated form compared to a conventional formulation of lopinavir at 400-100 milligrams. You can see that despite the 50% reduction in the lopinavir dose, the GMR, the geometric mean ratios here are within 0.8 to 1.1 across the C trough concentration, the area under the plasma concentration curve, and the C max, indicating that we have similar pharmacokinetics despite the 50% reduction in dose. Similarly for efavirenz, again a 50% reduction to 300 milligrams compared to the clinical dose of 600 milligrams. So we had equivalence in the area under the curve. We had equivalence at a 12-hour concentration at C max. We didn't see that at the C trough concentration, but I would highlight that this is because the C trough concentration was actually higher for the lower dose nanoformulation than it was for the current clinical formulation. Graphically, the data are summarized here, so you can see the clinical formulations shown in black. You can see the nanoformulations shown in blue, and we did get this indication for both drugs that we were extending the tail, indicating you had a longer duration of exposure for the nanoformulations. So there are some limitations. I'll just run through the limitations. It's a low sample size, obviously. N equals 5 for lopinavir, N equals 4 for efavirenz. There was no direct comparison with the clinical formulations within this study, so it relies upon historical data for the comparison. But we are now moving into a stage 2 analysis where we'll put the nanoformulations head-to-head with the current clinical formulations. Dose prediction above the study doses, so particularly for efavirenz where we extrapolated 50 milligrams to 300 milligrams, does assume dose linear pharmacokinetics. But I would point out that for efavirenz, this has been demonstrated across adult doses in previous studies. So to summarize, both the solid drug nanoparticle formulations proved to be well tolerated at the studied doses. There were no grade 3-4 adverse events. The data confirmed the potential for a 50% dose reduction while maintaining therapeutic plasma concentrations of the drug. If confirmed in future studies, our collaborators at the Medicine Patent Pool in Geneva have estimated that the approach can save up to $243 million US dollars per year, while also freeing up manufacturing capacity up to 930 metric tons per year. Very exciting in the context of deployment of these formulations in low and middle income countries. We do need to do further formulation development for future translation. We're looking at co-formulation, tableting, and improving stability. But the approach has wide applicability to drugs from several classes, and we have a number of programs ongoing now for other antiretroviral drugs, as well as other diseases for oral and long-acting delivery. One of the programs we're currently working on is the Optimize program. It's funded by PEPFAR through the US Agency of International Development, and it integrates a number of key collaborators to try and bring down the time to get into the market with these types of formulations. Most of our preclinical expertise is available through the Uncl. So if you missed the Uncl session yesterday, then there's a stand outside. So I would encourage you to interact with us through the Uncl. I'll leave you with some thanks. The team in Liverpool, Steve Ranard, who invented the formulation approach, Marco Siccardi, who does all the physiologically based pharmacokinetic modeling, Marta Baffito, who designed the clinical protocol for this. And I can't thank the Clinton Health Access Initiative enough for the regulatory support they've given us to get to this stage of development, and also the Medicine Patent Pool. And I'm happy to take any questions.
Thank you very much for this important presentation. Perhaps I could start off with a question. When you do physiologically based pharmacokinetic modeling, are there any principal differences between adults and children?
Absolutely. So the approach we used here is a population PK model. So it's fundamentally different to the physiologically based PK modeling. So the PK modeling takes the actual clinical data and fits the model to the clinical data. Physiologically based PK modeling will take in vitro data and try to model the pharmacokinetics, so it can be thought of as an in vitro to in vivo extrapolation. And I say the word we, Marco Siccardi's done a lot of work around pediatrics with physiologically based PK modeling, and we have a number of papers in the literature which describe the approach. And there's lots of things captured in that. There's obviously differences in drug metabolism, enzyme expression, as well as a number of other anatomical differences which are captured in the modeling approach.
Very good, thank you. There is time for a few more questions. Yes, please.
Andrew, many thanks for a talk in a very interesting and very important field of therapeutics. I'm aware of previous research and applications of similar drugs, or also antiretroviral, implicating hot melted lipid tablets. Can you make a comparison of the advantages of your formulation compared to this one?
So, come back on, yeah. So actually the lopinavir, the current lopinavir clinical formulation is made by that approach. So we've actually got a head-to-head comparison of the two, which is ongoing now. In terms of the actual process, there are benefits in terms of cost of the approach that we're using. I didn't go through it in detail here, but we essentially just make an emulsion of the drug and excipients, and then we simply spray dry it. So in a single step, you get formation of a powder, which has nanoparticles embedded in a matrix of excipient. So in terms of speed of process, cost of process, and compatibility with excipients from the FDA CEDA list, we have benefits across those.
I think it is time for us to move on, but let me thank the speaker once again.
Thank you.